Hello students, today in this video we'll be covering a Lebanese biology official exercise for grade 12 SE about transgenesis. Please pay attention to read the given carefully, and the given includes the title, the text, the figure. I recommend you circle the important terms, and when starting to read the question, read the whole question, understand the objective, and pay attention to the action verb. So this is our official exercise. Take some time to look at it and think of it. So let's do some recall before we start solving an exercise, starting with the word transgenesis. What is transgenesis? It is the process where we introduce an exogene. This exogene could be from a different species or a different organism of the same species, and we allow this organism or the recipient to have a new phenotype, and this phenotype is transmitted to its offspring because the gene will be transmitted to the offspring. So three main ideas for it to be transgenesis. We have an exogene, and this exogene is providing a new phenotype, and this phenotype is transmitted to the offspring. Some application of transgenesis in agriculture. We produce plants that are resistant to insects. We produce plants that are resistant to certain diseases. We can improve the production of a certain species. For example, we can facilitate farming in certain harsh conditions, in certain type of soil, in certain type of weather, and we can increase the growth of a certain plant. We can enhance the appearance of plants, and even we can enrich the flavor of plants. It is also used to add the nutritive values to plant. For example, we can add certain vitamins to plant. We can even create vitamin plants that have vaccine inside them, so we call them edible vaccines. We need to remember or to differentiate between the donor and the recipient. The donor is the organism that is going to give me the gene of interest. The recipient is the organism that's going to receive the gene of interest integrated, يعني put this gene into its genome and acquire a new phenotype. In transgenesis, we talk about vehicle or ways of transporting the gene of interest from the donor to the recipient. We can use artificial vectors. We can use bacterial plasmid. Plasmid is a circular DNA naturally found in bacteria. We can use bacteria itself, for example, when we use agrobacteria to infect plant cell, and we can use engineered harmless virus. We also talk about a host cell or a copy machine or cloning, which is we have many copies of our gene of interest. And for this, we can use a bacteria where we reinsert the recombinant plasmid into a bacteria, or we can use a yeast cell where we reinsert the recombinant plasmid into the yeast cell, or we can simply use plants. So back to the exercise, we are having a loss in the corn plants, and that's because they are being damaged by the larval insects, specifically the larva of pyrale. To solve this problem, they use transgenesis procedure, where they produced corn BT plants that are resistant to pyrale larva without the need of using insecticides. So what happened is that they used bacterium BT. Bacterium BT has a gene that codes for a protein, which is an insecticide protein that kills insects. So what they did is the following: they extracted the plasmid of this bacteria BT, and they extracted the gene of interest after they done multiplication of the gene, and then they inserted in the corn cell. To insert the gene of interest in the corn cells, we have two procedures. We can simply use a gene gun, and yes, a gene gun is what you've just imagined now. It is a gun, and instead of the bullet, we are going to have the DNA of interest, and of course, the DNA is going to be coated. They use actually a gold coat layer to code the DNA of interest. What happens is that just as you press on the gun, this DNA is going to be inserted in the corn cell. The good part is that it's a simple technique. What's not very good about it is that we can't be sure that the gene of interest is being inserted in the nuclear DNA. It could be inserted in the mitochondrial DNA, or it could be inserted in the chloroplast DNA, or even it could cause modification to the corn cell, leading to problem in the corn cell. So the other technique is using agrobacteria with tumor-inducing plasmid. So here we're going to use another bacteria. This agrobacteria naturally has a plasmid. This plasmid is going to cause tumor into the plant cell. So by modulating this plasmid of the agrobacteria, here we're talking about a different bacteria, different from the Bt bacteria, we can modify its plasmid to prevent it from causing tumor, but still using it to infect the corn cell. So using this agrobacteria, we can infect the corn cell with the gene of interest. Then we are going to allow the cells to, we're going to culture the cells, putting them in a culture medium to produce more cells. If we look at this culture, we can notice 5 cells, 3 that contains this blue part, and the blue part is the gene of interest, while the 2 other cells don't contain the gene of interest. So to avoid growing cells that doesn't contain the gene of interest, we're gonna select the cells that contain the gene of interest. To do this, we can use antibiotic resistant genes. So while introducing the gene of interest into the corn cells, we're going to introduce another gene along with that gene, which is antibiotic resistant gene. And then we're going to grow all the cells in an antibiotic culture. Antibiotic is going to kill all the cells except for the cells that are able to resist the antibiotic, and these cells will have the gene of interest along with the antibiotic resistant gene. So we will end up having or growing only the cells that contain the gene of interest, or we can say the transgenic cells. Of course, this culture medium is going to contain nutritive material and plant hormones to allow growth of the plant cells. So we will end up having corn Bt plants that are resistant to the pyrrole larva. So solving the questions, we have to indicate is a simple answer. I need to indicate the cause that leads to the loss, and I need to indicate the donor, and I need to indicate the receiver, and I need to indicate the enzyme involved in extraction of the gene of interest, and then I need to justify why will I consider these corn Bt plants as transgenic. So the cause of the loss in the corn plants is damage by larval insect known as larva of pyrroles, and the donor is the bacteria Bt, because I don't say the plasmid, the donor is an organism, and the organism in this case is the bacteria, because the bacteria over here is providing the gene of interest, which is in this case a gene that codes for a protein insecticide. Who is the recipient? The recipient is the corn plant. What is the enzyme that I use? I use restriction enzyme. Restriction enzymes are enzymes naturally found in bacteria. They are the biological scissors that are going to cut the DNA in precise places. Why would I consider this Bt plant as a transgenic? Because it contains a foreign gene, and this foreign gene was isolated from a different species from the bacteria, and it was integrated into the DNA of the corn plant, allowing the corn plant to have a new phenotype. The new phenotype here was being resistant to insect, and this was transmitted to the offspring. We noticed that we have many plants that are Bt resistant plants. So whenever we are asked to prove if this organism is transgenic, we're going to focus on these ideas. It should have a foreign gene, also called transgene or an exogene, either from a different species or from a different organism from the same species. It's going to produce a new phenotype, and this gene is transmitted to the offspring, so the offspring will have the same phenotype. Now, the second part of this exercise is the following, so please read the question. Okay, so we are checking the mortality rate of the Pyral's larva, and we're checking this mortality rate in two types of corn plants: a transgenic corn plant and another non-transgenic corn plant, but with that was treated with an insecticide. So we're seeing how many Pyrals are dying in these two different plants, and the results are shown in the document here. The question is, show that using transgenesis to fight pyrals is more effective than using insecticide. And then the other question 5 is, justify why cultivating corn BT plants contributes to protection of the environment. To start with, shows that is an action verb where we have to use logical reason and then answer. And when using the logical reason, the best way to use logical reason is to link the cause to its result and try to find a relation between them. To do this, we can use compare and analyze, or compare or analyze. So what we did is the following, or the answer is the following. We've noticed that the mortality rate of pyrals is 100% in transgenic BT corn plants, greater than the 70% in the non-transgenic corn plant treated with insecticide. 100 is greater than 70, so this shows that transgenesis is more effective than using insecticide. Instead of this, I could have used the analysis term, like I could have used the mortality rate is 100% in BT corn plants. This rate decreases to 70 in non-transgenic corn plant treating with insecticides. So this shows that transgenesis is more effective than using insecticides. What are the benefits of using transgenic corn plants? First, they're going to ensure better protection of corn plants. We've noticed that 100% of the pyrals are going to die, so all the pyral larva is going to die. So I'm going to ensure a full protection for the corn plant. And because I'm not using insecticides, and because insecticides are pollutants, so I will be reducing pollution. So transgenesis in this exercise is beneficial for two ways, or for two things, because it protects more corn plant, and because it avoids pollution. Thank you so much. Hope you enjoyed this video. Stay tuned, and there are going to be other videos explaining more about transgenesis. Stay tuned.